So I will now present some recent applications. For example, applications to Huntington's disease. And also applications involving the Allen Brain Atlas, and then some exciting applications about human pre-implantation development and Alzheimer's disease. So by now, you of course know what is WGCNA. It goes through these steps. You know that it's used for reducing high-dimensional data and integrating multi-scale data. And to highlight this aspect that you can reduce complex data, I now show you a recent application from 2012 where researchers at the Allen Brain Institute created a very exciting data set. So they collected brains from 2 healthy males, ages 24 and 39. And so in certain ways, the sample size is 2, okay, 2 brains. But they were very careful of measuring actually 900 microarray samples per individual, and they queried 170 brain structures. In other words, they chopped up the brain into tiny cubes and measured expression levels in each cube. And they used an Agilent microarray that had 64,000 probes. And so these data are freely available. You can download them, and they provide a neuroanatomically precise genome-wide map of these transcript distributions. But here, the authors used WGCNA in order to see what are the main themes, you know. So they looked at all 900 microarrays from the first human subject, brain number 1, and applied WGCNA and found a very clean cluster dendrogram, you know. Remember, these are 64,000 probes, you know, but they can be clustered into really a handful of modules. Now, the other thing you can ask is, well, what is known about these modules? What do they correspond to? And again, one strategy is to plot the module eigengenes versus different brain structures. For example, module number 1 corresponds to primary sensory motor cortex. Why? Because its genes are overexpressed in these regions. Module 6 corresponds to striatal genes. You know, notice the module eigengene takes very high values here, but is 0 essentially everywhere else. But other modules correspond to cell types. For example, module 8 corresponds to microglial cells, module 10 to astrocytes, and so on. Module 12 to oligodendrocytes. How do we know it? Well, we study module enrichment with respect to the cell markers, you know, user list enrichment analysis. Now, you know, of course, how we construct the weighted correlation module. And I mentioned to you also consensus module analysis. But now I wanted to apply it to Huntington's disease data. Actually, the analysis was again done by Peter Langfelder. And he downloaded 2 publicly available data sets from these publications. And there were, for this data set, there were multiple brain regions. But we were interested in the caudate nucleus area. Why? Because Huntington's disease, which is a neurodegenerative disease, affects the caudate nucleus. And so we applied the signed WGCNA and identified consensus modules and then related them to Huntington's disease status. And look at this. In essence, we get 2 very large modules, the turquoise module and the blue module. There are other tiny ones, but these are the main themes. The next thing you can then ask, well, how do each of the genes relate to Huntington's disease status? And you see brown, meaning overexpression, and blue, underexpression. So in certain ways, this module analysis is trivial. There are two modules. One is comprised of genes that are highly overexpressed in diseased subjects, and the other is underexpressed. Now, to a biologist, that is not surprising because Huntington's disease is a neurodegenerative disease. It kills off medium spiny neurons. There are vast differences in the brains, and that is reflected in this clear signal. So what we could do then with the module eigengenes, we related them to Huntington disease status, but of course you can relate it also to the Huntington disease gene. Some of you may know that Huntington's disease is a trinucleotide repeat disorder. There's a Huntington disease genes, and the more CAG repeats you have, the higher the risk that you get the disease. And so here we relate then these modules that we found to Huntington's disease status. And notice the turquoise module is highly related to the disease, and as is the blue module, p-values 10 to the minus 10. Now, using the user list enrichment analysis, we found that the blue module corresponds to astrocytes. And so what's going on? Well, this module corresponds to immune cell reaction. The neurons are dying. And so that's one aspect. The other module, module number 1, corresponds to neuronal genes. And so what do we learn? Well, we find what we know. Neurons are dying. But it's an old story. However, we learned it purely based on gene expression data. If I had known nothing about Huntington's disease, I would have been able to tell you this is a disease that involves the loss of neurons in the caudate nucleus, and there's an immune reaction, just based on gene expression data. So again, it matches the neuropathological story. Now you know also all about module preservation by now. And now we can use these module preservation statistics to address an interesting question. Remember, the modules were defined based on the caudate nucleus brain region, but there are many other brain regions. And you can ask, well, are these modules preserved in other brain regions? For example, Brodmann area 4, Brodmann area 9, or the cerebellum. And also remember, our most important modules were the blue and the turquoise module. And what you see is that the z-summary statistics for these modules are high. So yes, these modules are preserved in the different brain regions. And why is that a reasonable statement? Well, remember, these modules relate to either neurons or astrocytes. And so they measure in certain ways cell type abundances. And of course, you find these cell types also in other brain regions. So it's very natural that these modules are preserved. But it's nice to observe it statistically with a z-summary statistic. You can, of course, also correlate the modules, eigengenes, to disease status in these different brain areas. And then the story becomes more complex. So the turquoise module does relate to disease status in Brodmann area 4. Why do I know it? Because it's statistically significant. This is a z statistic, so it lies below the threshold of 0.05. But the module eigengene of the turquoise module does not relate to disease status in Brodmann area 9, right? So meaning that we know the module is preserved in Brodmann area 9. It exists, however, there it does not relate to disease status. So disease doesn't affect Brodmann area 9. That's one way to look at it. And again, this matches what neuropathologic studies have shown. But we learn all of that based on gene expression data. So, of course, the future use of these module preservation statistics for HD is to now relate these human disease-related modules to mouse models. There are many wonderful mouse models of Huntington's disease, and we can just ask, well, which of them matches human data? But also we can ask things like which of the brain-related modules can be found in human blood tissue, because it could, if we are lucky, lead to surrogate markers, and biomarkers of the disease, although this is a very much speculation. Let me now come to another application that may be interesting to some of you. So this application was carried out by my colleague Guoping Fang, Kevin Huang, and many other authors. And they had a very precious data set where they used single-cell RNA sequencing, right? As, on individual cells from human pre-implantation embryos. Just to remind you, you have of course learned a lot about RNAseek data, but they are typically based on large tissues. What is special here is that Guo Ping and his colleague from the Beijing University, they sorted cells and measured RNA-seq in individual cells. And to give you a sense what the data look like, and to review pre-implantation development, remember you start with an oocyte. And so there were 3 oocyte cells from humans. And then it goes to pronuclear cells and zygotes, and then you get a 2-stage and 4-stage and 8-stage, and then the morula, and finally it implants. Again, I'm just a statistician, but, but in any event, when you do a principal component analysis of these data, the RNA-seq data, you really get this nice patterning, which already tells us that the data are meaningful, But now we applied WGCNA to these data, and we found a cluster tree with very distinct branches, indicating, again, high-quality data. And now what we can do is we can plot the modules versus these different developmental stages, meaning from oocyte to morula. And now, different from our conventions, the genes are columns. So for example, here's this BISC module, BISC4 module, and all of its genes are highly overexpressed at the oocyte stage, and then they are underexpressed in later stages. And that's why we call this module the oocyte module, Which, let me digress and mention this. When you publish a paper with WGCNA, don't mention in the abstract, we found the BISC module, or the maroon module, You want a more biologic name. You want to say, this is the oocyte module, right? Or the oligodendrocyte module. As the author of the article, you would want to come up with a better name. So in any event, many of these modules that we found really correspond to different stages in pre-implantation development. And so it's very exciting, that the transcriptomic data recapitulate. So now you can look at which modules contain certain transcription factors. You do get lucky because some transcription factors correlate highly with the expression data, Some don't, but some do. And so you can still learn a lot, Here we show how the module eigengenes, remember these are RNA-seq data, so these are PKM measures, and we plot here the median RPKM across the different stages, and as I said, the BISC4 module is highly overexpressed in the oocytes, and then it continues to go down. And so it gives you a sense how you can visualize module eigengenes versus different categories, That's often what we do. Now, we also related these human pre-implantation data to corresponding data collected by other authors. For example, using the standard microarray platform. And so And so we needed to convince the reviewers, of course, that this module is robustly defined. And so here we show them these, the Z-summary statistics of our modules. And what you learn is this oocyte module, for example, is highly reproducible in the data from Vaseva, in the data from Xi, and so on. But we not only had human data, but we also had mouse data, you know, so we can ask which of these modules are highly preserved in corresponding mouse data. And yeah, so overall, many of the modules were highly preserved, especially the stage-specific ones. Remember how I said many of the modules are stage-specific, but some are not. And it is the modules that are stage-specific. They are highly preserved in mice. All right, let me now come to, do you have any questions at this point? Or? No. I mean, I like the application a lot because it shows you these methods will be also very valuable for RNA-seq data, no question. I know it because many of my colleagues use it. And several people have asked me, do you have any problems with the fact that these are count data and not intensities? And we really don't. You know, the correlation measure is a very appropriate measure for relating counts, you know. Yes?
So just to be clear, the data sets you're getting from these other papers,
yes.
Are they all including expression data, or could you just take a list of genes?
No, no, these are expression data.
Okay.
Because
so
we
can
can.
take just genes and correlate it.
If I just had lists of genes, I would use just overlap. It's a cross-tabulation approach, you know, which is a good approach, you know. Notice we do that here. I didn't talk about this panel, but here we actually look at overlaps of genes. How many are shared between pairs of modules, you know. We have R code for looking at overlaps, you know, so send me an email if you want it, yeah.
So speaking of RNASeek data, so one of the problems with RNASeek data is generally the data sets tend to be smaller.
Yes.
So your this network approach from what you said seems like it's much more robust if you have more samples. So where do you kind of, at one point you said so if you don't have at least 20 samples,
yes.
it's hard to build a network.
Yes.
So, I mean, most of the RNASeek data sets I've seen are many fewer than 20 samples.
Yeah, then I mean, we've written papers with samples sizes of 15 or so, you know. When you have very precious data, you have to just do what you got to do. For example, the chimpanzee brain data were very precious and very rare, you know. It's, and I don't remember what the sample size was. I want to say 18 or so samples. You know. But it is true, if you have fewer than 10 arrays, please don't use these methods, you know. Yes?
In your first slide in this section, you showed the principal components where you actually
yes.
separated out the stages into
yes.
basically 3 different dimensions.
Yes.
How, what did you learn by going beyond into looking for modules? Is there a lot of information here that tells you about the various stages?
Well, I mean, I like this plot a lot because it shows how the samples are related. And so, but if you wanted to be critical, you would say, I learn nothing because I know already that oocytes precede pronuclear, precede that, you know. And but it's nice that it's reflected in the expression data, you know. But fundamentally, we don't learn much biology here. That's very different for the WGCNA analysis because you can now say, wow, I have a set of genes that are oocyte specific, you know, they really precede then the pronuclear phase. You can ask, what are these genes? Which transcription factors regulate them? You know, so you can learn a lot of biology by looking at the genes that are part of this module and also hub genes in that module, right? You would now calculate the KME measure And you would say, well, what are the hubs here? You know, do they happen to be transcription factors? You know, and so this partitioning of the data yields a very valuable annotation of genes. You know, imagine people who study birth defects. They could now say, well, does my favorite disease relate to genes in one of these stages, you know, where something could have gone wrong, just to wildly speculate. Yeah. Yes?
Samples again were each stage here?
Yeah, look at this. So in the first oocyte stage, we had 3 human samples. And here I want 5. You see? I'm not good at counting it, but it was enough to do the analysis. But the other, yeah.
As long as there are enough samples overall, you feel that that's okay. It's not
Yes.
a matter of samples per group as much as a matter of how many samples you have overall.
Yeah, that's true. But also the other thing is, it all depends on noise level, right? Because here this is single cell RNA-seq, and arguably these collaborators were incredibly careful, and these are very high signal data. I can say this, make this statement because I see very little noise. Do you notice there are hardly any singletons, you know? Very distinct branches. So to me, although this is not a rigorous statement, but to me it already means there's relatively little noise. Somehow these genes, and also it means the heat map tells you the story. Notice there are vast changes. There's very high expression in the oocyte stage, very low in the later stages. That's also captured by these box plots. Look at the 8-cell module, very high at the 8-cell stage and low everywhere else. So there simply is a very large signal in the data, and so you get away with a smaller sample size. Yes? Oh, no. Any other comment? Yeah. Okay. So everything I talked about really could be considered systems biology. We learn biology by looking at modules. It's a systems level analysis. But there's also something called a systems genetic analysis. And so let me outline some approaches for that. So I'll talk about a software which is called the network edge orienting software, which allows you to do causal inference based on genetic markers. And so let me mention it here. So the overall question is, can we use SNPs for learning directed networks? What do I mean by directed? Well, there are directions. This causes that. And there's actually a vast literature on that topic. So yes, you can use SNPs to infer causal directions as long as you are willing to make certain strong assumptions, you know, which are debatable. But these approaches make use of this fundamental paradigm of biology, which says sequence variation precedes changes in messenger RNA abundance, which then leads to changes in proteins, which then may change things, may affect clinical traits. And in this flowchart, there's one thing that is irrefutable, which is that sequence variation. In other words, for example, SNPs precede everything else. So the SNPs can be interpreted as causal anchors of the analysis. So in other words, if you find a significant correlation between a SNP and a gene expression value, then it's not the fact that the transcript changes the SNP, okay? It has to be that the SNP variation affects the gene expression. So this is a causal anchor, therefore, this one direction. Now, assume we have the following situation. So we have expressions and we have HDL. And what we know is that these data are correlated, but notice there's an edge without an arrow. We don't know what causes what. However, we further assume that we have SNP the SNPs that are represented on these chromosomes. And so if a SNP is correlated with, for example, insulin level, it has to be that changes in the SNP affect insulin level. It's not that if you increase the insulin, your genetic, your DNA changes, right? So now we use then these genetic markers to arrive at an orientation. That's the idea. So we get edges, and you notice that the edges have scores, which are called Leo scores, local edge orientation score. And if you have a high Leo score of, say, 3.5, then you say there's strong evidence that this gene causally affects HDL levels. Okay. Now, so the NEO software then takes as input a number of quantitative variables. So it could be physiologic traits, could be gene expression data, and it takes as input genetic marker data. And then it outputs these Leo scores for edges. Now, there is a software on our web page which you could try to learn. And if you run it, it outputs an Excel spreadsheet where the rows correspond to different edges. For example, gene A affecting gene B, and you get then this Leo score. And then you would say, well, actually this Leo score is high, and you would say, I have some evidence, causal evidence, that there's a causal relationship between A and B. So the way these Leo scores are calculated is by using so-called structural equation models. So what are these? I hope many of you or most of you know multivariate regression, just linear regression models. But statisticians have developed a generalization of regression models, and these models are called structural equation models. And so these models can be visualized by these different causal situations. So assume you have two traits, meaning gene A and gene B, or let's say gene A and this is insulin status. And then we have a genetic marker, a SNP. So then one causal scenario is that the SNP affects the gene, and the gene affects then insulin levels, you know. And there's a causal diagram, and it is what it is. But there could also be other causal scenarios. It could be that the SNP affects B and B affects A. Or it could be that the SNP affects both A and B at the same time. Or this or that. So there are different causal situations. And now what these structural equation models do is they produce a fitting index, a p-value, that measures how well your data fit a causal situation, you know. And so they could tell you, you know, this model, this causal model fits my data well, whereas the others don't, you know. Now, when you have multiple markers, you get more complicated models, but I won't talk about them. So there are certain Chisquare tests that assess how well a model fits your data. And these Chisquare tests allow you to calculate a p-value. But different from everything you've ever learned about p-values, you want to find a high p-value here, okay? That's what's the crazy thing, right? You always want a low p-value to have a statistically significant result. Except in the situation of structural equation models, you want a high p-value because it indicates that your data don't violate the causal model. The data, it fits well. All right, so as I mentioned, we have these different causal models, model 1 through 5, and for every model, you get a p-value. And in principle, if you want to decide which of these causal situations applies, you just pick the one that leads to the highest p-value. It's a very simple exercise. Now, in practice, when you have thousands of genes, looking at all the p-values is a bit tricky. And therefore, what we do is we define a relative fitting index where we remember how I talked about the Leo score. So we take the p-value for this causal situation that A causes B and divide it by the p-value of the remaining 4 models, the maximum of the alternative models. So, and then we take the logarithm of this. So the Leo score is a relative fitting index. If it is high, let's say larger than 1, it means that your causal fit of that model is 10 times better than any alternative causal scenario. So therefore you would say, you know what? I think this is probably the causal situation, you know. So typically, I actually only ever use this software when I have 2 traits or 2 genes of interest. I only applied it to 2 at a time, but you can generalize it, you know. And the software allows you to fit more complicated models. And so let me come back to this application that I mentioned earlier about hyperlipidemia. Familial combined hyperlipidemia is, of course, an atherogenic dyslipidemia, and it is characterized by elevated fasting plasma, triglycerides, total cholesterol, ApoB, and so on. And so, as I mentioned, my colleagues, Päivi Päiväkanti, had found one particular SNP using a linkage analysis. And now, so here's an opportunity. We have a very credible SNP, and we can use it as causal anchor. That was the idea. Use the SNP to do a causal analysis to find which genes cause hyperlipidemia. Why would that even be interesting? Because, of course, there are many genes that correlate with dyslipidemia, but you don't know, do they cause it or are they reactive, right? It could be that high lipid levels change the expression profiles of some genes, meaning the genes are reactive to the disease. And it's critical to distinguish which genes cause the disease from those that are simply reacting to it. So now in the original analysis, there were 870 individuals, but my colleagues only measured 70 extremely discordant individuals, right, where some were extremely healthy, low lipid levels, and half of them were extremely dyslipidemic. And so then they applied this WGCNA analysis. You've seen this plot before, and they found this TAN module, very related to the SNP. But now this module was enriched with relevant gene ontology categories, no surprise here. But now let's describe this network edge orienting analysis. So look, we could now assess whether this SNP causally affects the module eigengene, which then in turn may give rise to dyslipidemia. And when we did it, we found that at least for triglyceride levels, there was weak evidence, you know. See, a threshold of 0.3 is nothing to write home about, you know. It's not terrible, but it just isn't very significant. However, it's a much more meaningful analysis to do this on a gene-by-gene basis, right? Because the TAN module contained maybe 500 genes, and of course not all of them cause the disease. There will be some of them that cause the disease. And therefore, we applied this causal analysis on each of these markers, and we found 18 causal candidate genes for disease status, 170 for triglycerides, and 13 for both. So there were these 18 causal candidates for hyperlipidemia in that TAN module. And then my collaborators evaluated these genes, and many of them were actually known from the literature. But also, they then found that some of them co-located with SNPs that had been found in a GWAS study. So in this, so there was strong evidence from the literature that yes, some of these causal candidates are indeed causally involved with the disease. So we have a web page for the NEO software where you can download the software, but what I will do is I will briefly go back to the tutorial that we discussed earlier, the WGCNA tutorial. You've seen it before, and there is a section that we skipped earlier, which is called network edge orienting. Here it is. See, there's a whole part in the tutorial called Systems Genetic Analysis with NEO. And in this tutorial, I present some R functions for you, So you want to read in the SEM package from, forgot the author, I want to say Fox, but I apologize, I forgot the author of the package. In any event, there is an R function called Leo single anchor analysis. Why is it called single anchor? Because I have a single SNP here. I use a single SNP for causal inference. And then I can input the SNP, I input the module eigengene, and I input mouse body weight, right? And now I can ask, well, is the blue module causally related to increases in body weight? And the answer is no, not at all. It's a negative number. Do you see this? There's no evidence whatsoever that the blue module that we found in liver, quote unquote, causes a mouse to become obese. If anything, yeah, there is no causal relationship. It is what it is. So in this sense, it's a nice analysis. And here's some code for implying individual genes. Remember, the blue module contained hundreds of genes. And so then I asked, well, which of the genes in the module shows evidence of being causally related? And then I found these types of genes, and with a high Leo score, So this gives you a sense what you can do when you have a SNP. But I do want to caution you that the key input here is a credible SNP. You want to use SNPs that are extremely significant, meaning they were implicated in multiple studies. You really trust them, Because as you know, many SNPs turn out to be false positives, and so you can really use the wrong input, and you produce nonsense, And the other thing is, these causal analyses make several strong assumptions. And therefore, whatever you find with them, it really requires validation, So in certain ways, these techniques allow you to come up with good hypotheses, But then you would want to use some knockout studies or transgenics to really verify that, yes, the causal genes really do what you think they do. All right, I think I leave you with that. And if you have questions, always feel free to email me. I hope I can answer it. And I will still stick around for an hour, so if you have questions, please approach me. But unfortunately, then I have to leave, for the airport. All right, goodbye. See you at dinner. Yeah.